So hi everyone, thank you again for joining us today. We are here today with Ernest Stark, and he'll be presenting his latest work. I don't think he needs any introduction. The work today is Harmonic self-conditioned flow matching for multi-ligand docking and binding site design. Thank you so much, Ernest, for I think this is the first platform where you are sharing this work. So we are privileged and honored for you to be doing this, and looking forward to it.
Yep. I'm also looking forward to it. And back to the roots, presenting the original places. Very nice. Yeah, then folks, if you have ever any questions, just ask them. You know that I'm a fan of having more of a discussion rather than a talk. And yeah, today we'll be talking about this harmonic self-conditioned flow matching for multi-ligand docking and binding site design paper. And concretely, the task that we mean, or that we more or less want to address here, is this binding site design. And in the process of that, which we will call our model or our generative process for that FlowSite. And in the process of developing that, so to say, we will be looking a little bit at this multi-ligand docking thing and have what we call harmonic flow for that. No, that's not what I wanted. Okay, but then, oops, no, iPad Pro. Okay, so what we have here, right? Why do we care about this problem, or what is the inspiration to this? I guess most of us here know that basically every function of a protein or of a protein is carried out by interacting with other molecules, and some of those interactions are proteins with proteins or binding to another protein. But a large part of important functions are also binding to small molecules. And yeah, if we can engineer proteins to bind to small molecules, this can be interesting. And one case where this happens very often, that we have a protein interacting with a small molecule, and we sort of are have the protein perspective of engineering a protein, right? We've often seen in drug discovery the other side, that we have the protein given, and then we want to engineer this small molecule such that it binds to a particular pocket. But yeah, this other scenario, the other way around, maybe often occurs in enzymes. If we have a particular reaction of, like here, for example, we have this enzyme in blue catalyzing the reaction of the small, no, not of this heme, catalyzing the reaction of this arginine, which usually actually has an additional little thingy here. Let's draw that. Usually has its little nitrogen here, but in this image here, the nitrogen already wandered away to over here and formed this nitric oxide with an oxygen. And this reaction of this arginine plus an oxygen to nitric oxide is here catalyzed by this protein and this heme, which is in complex with the protein. So the protein's job here is to stabilize this heme and put the heme into position, put the arginine into position, and we hope an oxygen comes along, and then it reacts with the nitrogen that is here or was here to form this nitric oxide. Yeah, so we have this as a first step towards enzyme design. For example, maybe an interesting thing is to design a ligand, a protein that first of all binds all of these reactants in place in which they need to be. And with that, we're at the task of designing a protein that binds specific ligands. And this is maybe one motivation for that, is designing or having this as a first step towards enzyme design. But of course, there are also many other applications of designing small molecule binders. Like if you want to design an antidote that sequesters some toxic small molecules, for example. Okay, but then how do we concretely define our task? Well, we have as input our the 3D structure of the backbone of a protein given, and we have a like without side chain coordinates, right, and without residue identities, and we have the 2D chemical graph of some ligands, which we will call a multi-ligand if it's multiple ligands, as input. And what we want to predict are the residue types. So, or what we want to generate are the residue types that bind this ligand or these ligands strongly inside of the pocket. And for that, we also jointly generate the structure in which the small molecule will bind to the pocket, right? The structure we assume is unknown, and we jointly design or predict the structure of the side chains while we predict what these side chains will even be, right? Okay, and I'm now saying again, right? We assume that we don't have the structure of the ligand as input. There are some traditional methods, and I think there's now also ligand MPNN, right? Where you have where you need the bound structure of the ligand as input already. Okay, well then, how do we do this, right? I said in the beginning, as sort of a subtask to this, we will look at HarmonicFlow, which where we just have this task of we have the backbone of the protein pocket, and we have our ligands, and then we generate the structure in which the small molecule binds to the protein. And here we take this flow matching approach to it, where we have Prudencio, what's the question?
Yes, in the previous figure, I was wondering, why do you consider the heme as part of the ligand instead of a constraint of the input? I know the position can change, but like it's less flexible than the actual ligand, right?
Yeah, so one could also sort of maybe say there's a bond of the heme with the there's a bond of this heme with the protein side chain here, and then just have this as a torsion angle, maybe, for example, and then somehow parameterize the rest of the flexibility of this and try to have it, I don't know, move somehow else, like some internal coordinates or something for this, but I'm not sure. This might be a little bit problematic with this ring structure. But yeah, we could use this additional knowledge of it being basically attached here as what the question is aiming at, I think. And yeah, maybe you can specialize your model further like this, but here, like this is also a thing of that we wanted with HarmonicFlow, that we can just have arbitrarily many molecules and arbitrary types of atoms and ions floating around, and yeah, generate structures for all of this, and supervise with arbitrarily many, or train with arbitrarily many molecules and ions and cofactors in the pocket. Yeah, and if you now were to, but you could also just define this to be part of your pocket, right? And not give it as input here, and then you just have these inputs. Oops. Then you just have these inputs, and then only dock your nitric oxide and your arginine. Yeah, you can also do that. And now we're looking at this HarmonicFlow thing where we want to generate binding structures, and for that we're using flow matching. So I will now dive into a little I think very easy to understand flow matching explanation for 3D data, maybe, or specifically catered to generating 3D point clouds, so to say. And I hope this is a very easy and simple to understand explanation. And yeah, that's sort of the goal here. And with that, right, we're starting very simple. We have, what do we want to do? We want to learn an ODE that will take some data from a prior to, and then that will take data from a prior to, that will take samples from our prior to samples from a data distribution. And to do that, we use this flow matching print. Well, right, we can already do that with some priors, with very simple priors like a Gaussian. We can do that with diffusion models. And then in the end, we also end up with our ODE or SDE, whatever, with which we can integrate, right, where we can take a sample from our prior and then integrate it to end up with samples from our data. And now with flow matching, we're also achieving this and learning this, so to say, vector field where we have our prior distribution, which is here then represented by some samples from this prior distribution. And we follow some vector field that pushes this distribution forward and hopefully to our data distribution. And here we're now just have this visualization of a single vector field that stays constant, where like these points or these data samples from our prior followed this vector field and then ended up representing some other distribution. And this is then what we want to do with our vector field. But our vector field will actually be changing with time and not just a single constant vector field. Okay, and then we have our friends like Alexander Tong that showed us that one very simple way to learn such a vector field is basically by linearly interpolating between samples from our prior and samples from our data, and then somehow using these linear interpolations for training. And so all we need is access to our prior, all we need is access to samples from our prior and samples from our data, right? And no longer, as in diffusion models, do we need a forward process, do we need to define a forward process where we, for example, add noise to a sample from our data to then end up at a specific stationary distribution. And we can have whatever we want as the prior, and this doesn't have to be the stationary distribution that we get when somehow transforming samples from our prior with this forward process or with this predefined forward process. Yeah, and the super simple primitive explanation of flow matching, how it works, is we take a sample from our prior, we take a sample from our data, which we will call this x0 and x1, and then we linearly interpolate between those. And of course, this is much more flexible, this flow matching framework, right? And here we're just looking at one specific instantiation, which we're using for this HarmonicFlow, which really is very simple. And so we linearly interpolate here. If we have point clouds, literally a 3D interpolation, and say we have time, and then we randomly choose a time between 0 and 1, say 0.7. And then at this time, or at this fraction of the interpolation, we select our x. So our x becomes this. And then we just put this x into a neural network and ask this neural network to predict x1. Like where did this interpolation come from? Where did this interpolation come from, so to say, and what was the data for it? And yes, so usually if you look at this, you see this x1 minus x0 here instead of the x1, but we can just also predict x1 and then use this vector field that we're learning here differently during inference. And we know that predicting x1, or we've seen in our neural network parameterizations for 3D structures quite a few times now, that predicting x1 is kind of well behaved, or in diffusion models back in the days we called it predicting x0, right? Where x0 was, so to say, the clean structure, and here we now x1 is the clean structure, and that's why we are going with this parameterization of predicting the clean data instead of predicting a difference. Okay, and then this is all that we do during training. We just do this again and again. We sample from our prior, we sample from our data, we interpolate, and then we calculate this loss. And then what do we do during inference? Well, we start, right? And this is now again not the general flow matching framework and specific. This is just a specific example of flow matching, and we start with our a sample from our prior. Then we have our neural network that we trained before to predict x0, to predict x1, right? This is now xt, and then we plug this into our neural network, and our neural network tells us that this maybe is x1, or our vector field, if you want to call it that. So it tells us that this would be x1, and then we interpolate between x0 and the predicted x1, and take a little step in that direction, and we update our xt. And then we put that xt again into our neural network and ask it to predict x1. This is maybe the new x1 prediction, and we interpolate, take a little step in that direction. And we do that again and again until we end up with our final prediction, or with our finally predicted x1. And that's all. That's it. This you can train, or this one approach or one flow matching possibility that you can use, and it's just very simple and maybe sometimes more straightforward or more well behaved than training a diffusion model, because you don't have to write this this noise schedule, so to say. Like, right, in diffusion models you sometimes need to mess around a bit with your noise schedule, and here you just have this interpolation where you only have the single t that works well for everything that we've tried so far. And if you another thing that you probably sometimes do to generate this x here, instead of to generate the x instead of just doing this linear interpolation, you can also add a little bit of noise to it afterward. And then I recently learned that actually you cannot just add noise to it, like all the you cannot basically always just add noise to it, but if you're at x1, or if your time is 1 or your time is 0, then you would in principle have to add 0 noise to it. But in practice we find that you can also just keep a constant level of noise if you're at 0 or at 1 and train with that. Okay, but then we've now said how this works, and then here's a little visualization of what this then looks like, right? This inference procedure where we take our little steps. And
this is one question.
Yep.
So you're doing this, the time step you're choosing there, this is a constant.
Yeah.
delta T, and this works well. I'm a little bit surprised that you don't need some, I don't know, integrator, which is more specialized to solve this, that you can just use here.
So the question is this delta T constant?
Yes.
Yeah, yes, it is. And what would your alternative proposal be?
I mean, the delta T could like depend on how much the direction changes from step to step, so that you don't overshoot, basically. Because I would assume if the delta T is too large, then you don't really follow the integration path you intend to follow, right?
Yes. So this would then be a problem, right? If our paths aren't straight enough, and if our initial x1 prediction isn't good enough, yeah, we don't find this to be an issue, and right, it would be fixed if you take just a very small delta T from the get-go. But we actually take 20 steps here. So our delta T is 1/20. So it's rather big, and it still works quite well.
Cool. Okay, thanks.
Okay, so maybe we can ask Leon, do you use in your equivariant flow matching, do you use like an adaptive step size solver or something?
Yes, we use I think it's called Tropy5 or something like this. And then only if we get this straight integration path, then we use the fixed step size solver. And there we actually also take 20 steps. But the Rummel-Kutter method, for every step, we evaluate the vector field like at 4 points, roughly. But I'm not sure, maybe just doing it like you would be sufficient there as well. This we haven't tested.
Okay, so you've not tried an Euler solver?
No, we haven't tried this.
Why is the first thing that you try a Runge-Kutter solver and not the simplest?
Maybe from the background that usually this adaptive solvers were required for us, because if we get it a little bit wrong, then because we need to compute also the change in log probability, and then the weights might be off if our solver is not very exact. Yeah,
I think you also desire a lot more preciseness in the
exactly.
Like if you are only training a Boltzmann generator for a single molecular system and not generalizing across molecules. And okay, but then yeah, I guess this video is not playing. That's a little bit sad. Okay, imagine this molecule flying over here in a very straight path. Sorry about that. Good. Then we have a few little tweaks here that maybe you also can try in some of your 3D structure or molecule structure generation applications. So which are that we use this different prior, right? Because now with flow matching, we can use whatever prior we want. And then we use self-conditioning, which we actually find to help a lot. And okay, let's get to the last point. I'll make my comment about the last point later. Okay, but then let's start with this harmonic prior. So what we think why this is especially nice for our problem is more or less if we're dealing with multi-ligands, like we have multiple molecules in our ligand, which if we sample the 3D positions of it in our Gaussian prior, right, then we just have our atoms randomly assorted around. But if we now use this sort of harmonic prior, then the stuff that is bonded together, that is initialized close together, and you sort of don't have to the generative process does no longer have to pull these apart, more or less, and resolve which atoms are bonded together or which atoms belong to a molecule. Okay, and of course you can also choose, for example, an RD-kit ligand initialization, I would say. Like you could initialize every molecule to be an RD-kit ligand or a structure generated by RD-kit, and then place them randomly, their translation randomly and their orientation randomly in space, and then start from that. And yeah, please try it. Try things like that if you're interested in that. And the one concern with that, right, could be that then you sort of have to maybe reason about, or what is then your generalization capability, because you have to reason, or does the flow then have to reason about how RD-kit, what are the outputs of RD-kits, and yeah, will it have to understand basically the rule-based system of RD-kit for generating these structures, or then knowing where to flow its stuff? But Prudencio, you have a question.
Yes, so I was just wondering how you, for some of these things, they should be close together, right? And how do you avoid kind of having infinitely large distances between some of the disco ligands?
Do you mean these distances?
Yes, yes.
That they are not too large? Well, this is just your standard deviation, as in a so the way that we sample this. Okay, let me talk a little bit about this this harmonic prior. So basically, the prior is proportional to this Boltzmann distribution, where the energy is defined by you have high energy if something is bonded and far apart. So everything that is bonded, like if you have a bond between I and J, then you want I and J to be close together. Otherwise, your energy is high, and then this thing with its minus in or exponential is unlikely if our energy is high and the bonded stuff is far apart. So, and the way that you actually sample this in practice is that you have your Gaussian or you have your X. Yeah, you have your Gaussian where your standard deviation or your precision matrix, yes, your precision matrix is just the Laplacian. So in your exponential of your Gaussian, you would have this. And so the Laplacian, which on its diagonal has the degree of each node, and on the off diagonal you have whether or not the stuff is bonded. Like if you have a bond, like the graph Laplacian is what I mean, right? And yeah, that's what we sample. And now if you have your Gaussian distribution, you can also just have a different standard deviation. Then you just no longer have ones all over, but different values. So then your standard deviation will control how close or how far apart our molecules are.
Okay, perfect. Thank you. There is a question by Elena in the talk. V theta can predict S1 directly from S0. Why do we need to have multiple time steps instead of using the predicted S1 directly?
Yeah. Why don't we just sample from here and then directly generate this? Well, so this whole process here, right, is only correct in the or when do we have the generative model that actually transforms samples from our prior perfectly to samples from our data? That we have if we really have the correct vector fields, or we have infinitely much training data. And so if our vector field is perfect, and and if the ODE that we're integrating it with infinitely many steps, or we're like continuously we're in the continuous limit. And here this is just a discretization of this integration. But how to better explain? I don't think that was now too useful. So let's do it this way, very intuitive, right? If we just take the oh yeah, we saw this. Right, our x1 prediction here was actually very bad that we had, and we had this sort of process, and this is the x1 that we predicted in the end. And sort of if you now have your generative model where you take only a single step, then you're like, right, because you have this continuous process, which maybe if you integrate with infinitely many steps would end up over here. And if you only took a single step, then your approximation error of this continuous process would be much worse. Yeah, I hope that helps. I can also maybe later on at the end switch to a video where I show sort of what the first x1 prediction looks like really, and then what the following x1 predictions look like. And we see the x1 prediction improving more and more. And sort of why don't we just train a regression model that takes some input, like some initialization, and then predicts x1? Well, that's because then we can't resolve, like we want a generative model to, for example, resolve asymmetries symmetries in a molecule. If we have a molecule like this, like a 6 ring, then the best thing that a regression model could predict is a line, right? Because if you're trying to minimize your where this atom is here and this instead of having the atoms here, because if you're trying to minimize you have this as your training target, and now you predict something, and if you're trying to minimize your mean squared deviation, and you have no way to distinguish this place between this place because you have no sort of randomness in your process, then you have no randomness in your process, so you can't make a decision between this place and this place in your symmetric setup. Then the best thing you can do for both of these atom locations is to put them both in the middle to minimize the mean squared error that you would have, because you can only make a single prediction, right? Okay. I spent a long time on that question, but maybe it was helpful. Okay, and this was our harmonic prior. Yeah, and now we come to our self-conditioning, which is also a very simple trick, but seems to help quite a bit. So, right, we said that during inference, we have our xt, and we always have our xt as the input to our neural network to predict our x1. And now we just say, no, instead, we additionally, as input to our neural network during training, during inference, we take the previous x1 prediction. So the xt that we predicted, the x1 that we predicted when we were over here, this one we also take as input to our neural network, and then we predict a new x1 from that. Yeah, and this of course then requires training a little bit differently. This requires training a little bit differently, and the way it's done is very similar to what we see in or what we know from AlphaFold's recycling. And this is sort of the inspiration for why we want to why we want to do this sort of self-conditioning, right? Because we know that from AlphaFold that recycling works pretty well, and predicting X1 works pretty well, or it can predict X1 pretty good with its recycling. And the thing we do is, right, we always feed our initial prediction back into the neural network. And then we sort of have at no additional cost, or almost no additional cost, during training and during inference. Well, actually no additional cost during inference. We have a much deeper neural network that here in our case has this many layers, or the number of layers that we have times the number of integration steps that we do. And in recycling, right, the way we would train AlphaFold is we do our recycling, but then we only backpropagate through the last recycling step, so to say. So all of these previous steps are not in the computation graph, and there's no additional memory cost and almost no additional time cost, because the backpropagation is usually what takes the time. And then here the way that we train this as well is that 50% of the time the model gets as input a sample from the prior, and 50% of the time the model gets as input an output of the model itself after the model had a sample from the prior as input. And just like recycling is trained. And then we can use this during inference. And this is a somewhat hacky trick, I would say, but it seems to work. And this trick doesn't come from us, right? This was also already used in diffusion models, and it comes from analog bits first, and was also used in RF diffusion or frame diff by Jason and Leon. What's the question?
Yeah, I'm wondering if I got this correctly. So now this ODE is no longer invertible, right? We cannot go from P1 to P0, can we?
Yeah.
Okay. So it's not possible, for example, to evaluate the likelihood of samples generated of samples. Sorry, not of samples, but of data points.
Yeah. So we can calculate the likelihood of going this direction, yes. But yes. not the other way around. Wait, okay. Is there a way to do it the other way around? Yeah, but I mean, in the end, just have now tuples, right? Instead of. Yeah, I'm not sure. I can't figure it out right now.
Yeah, but it's probably not often necessary to do so anyways. But okay, yeah, thanks.
Yeah, well, for you, would it be interesting?
Actually, it's for us, it's enough that we can evaluate the likelihood of the samples we are generating. I think it doesn't matter too much the other way around.
Okay, well, then we lastly get to our tweak. I'm not even sure if we should call it a tweak. It's just people, right? When people were using tensor field network, or we have these tensor field networks, for examples, and we have our equivariant graph neural networks. And with equivariant graph neural, like tensor field network is basically also an equivariant graph neural network, right? But with equivariant graph neural network, I now just mean this architecture by Satoras et al. And where we look at distances between edges and then update coordinates based on these distances. And in EGNNs, we would always update coordinates after every layer, right? And what people usually did when using tensor field networks for predicting some point clouds, as far as I know, is, or at least also what we did, and then found that, yeah, this sometimes didn't work so great, is to just predict coordinates. So we take some coordinates as input, and then we predict some coordinates as output. And instead, what we find to work much better is to also update, like to just predict updates to the coordinates after every layer and apply them to the coordinates and feed them back into the next layer. And then you like sort of have a little stub gradient for the non-differentiable parts going on in here. Yeah, and then we have this auxiliary loss to supervise each layer's coordinate prediction a little bit as well. Okay, and that's all that's happening here with these equivariant refinement tensor field network layers. But we really find that predicting, we find that first that tensor field network layers are, for many applications that we've tried at least, quite a bit better than equivariant graph neural networks. And then especially if you're now predicting structures, then predicting updates to your coordinates and moving them after every layer works quite a bit better again than just predicting coordinates at the end. And yeah, this is where we can already see this, for example. And here in this down here, we have basically this harmonic flow architecture with everything that we just discussed. And then here, for example, we have the performance, or okay, so I should say, right, what are we looking at here? We're looking at the percentage of predictions with an RMSD below 2, of ligand structure predictions with an RMSD below 2. And we're not using any filtering model or any confidence model, like for example in DiffDoc. Yeah, and then we have our standard tensor fields network layers, as we call it here, which just means that we don't do this these iterative updates, but just predict new coordinates. And this gets quite a bit worse fraction of correctly predicted structures. Yeah, and then we also have this velocity prediction thing here, where we where what we do is that we remove the what we mean with velocity prediction is just that we now no longer predict x1, but we predict x1 minus x0 as you usually do it in flow matching, so to say. And then we have our no self-conditioning here. So we also see that self-conditioning helps a bit. And we have our, as another little thing that we look at, right? Because the harmonic prior is actually something that we can use with diffusion models. It's not like it becomes quite complicated with a harmonic prior, but you can use it with a diffusion model. And have this harmonic diffusion process by Bowen, and we have a little comparison with that. Yeah, and so all that is to say, maybe you want to try these things if you do some flow matching stuff. Okay, and yeah, we also have a little comparison here where we have harmonic flow, right, with just this very unstructured thing of just predicting coordinates, and compare it with this sort of product space diffusion process where we have a diffusion over rotations, translations, and torsion angles. And we're doing this pocket-based comparison where we have some smaller networks than we, for example, have in the DiffDoc paper, and just keep all the parameters the same. We again have no confidence model or something like that, and we just look at the performance that we get on some different splits, and also in the blind scenario on the time split. Okay, but then maybe this is also promising for exploring this harmonic flow further for actually doing docking, right? And you could maybe push this to have a large model, to also build a confidence model and really tackle the docking task with it. But that's not what we're doing here. And also you have to keep in mind, right, that here, this is basically, if you now generate 10 samples, or let's say you generate 40 samples with your generative model, then this is now the average that we're looking at, like the average percentage of predictions with RMSD below 2. But if we have a confidence model that can find the best of these 40 predictions, then it becomes important what is the diversity of predictions that we're having here. Because if our harmonic flow is always to just predict the same thing, which maybe with our X1 parameterization, with the self-conditioning, could be encouraging that to have less diversity. And with the product space diffusion actually has something diverse. And then we have something that can pick out these the best samples from this diverse set of samples. Then this advantage here, for example, in the blind docking scenario might disappear. But right, in the end, we're thinking about this joint design scenario. And in the joint design scenario, maybe is kind of valuable to always have this precise structure estimation, and also a precise X1 estimation throughout your generative process. And yeah, we're actually interested in the evaluation metrics of the design and not the structure. Okay, but if folks want to explore this for docking, please let me know. And yeah, this I think might definitely be interesting. Okay, but then Michael, your question disappeared. Maybe you can get to it later. Otherwise, right, we now looked at this harmonic flow thing, where we just generated binding structures. But what we were actually interested in was predicting these discrete residue types. So now we will very quickly, because it's really super simple, and this is sort of what I like about this FlowSite setup, that it's a very simple setup for generating discrete and continuous data at the same time. And the way I will explain it is by first again going over the structure generation and putting it into this language, right? So we have in blue our contact residues, of which we want to predict the identities, and we have our harmonic prior multi-ligand initialization, so our initial structure for the ligand. And then in our structure generation, right, we used our neural network, our vector field, to predict X1, and then we interpolated with using our X0 to get this XT. And then we end up with this XT over here, right? And then we do this again and again and again until we end up with our final structure. And now what we do instead is that we, oh, right, and I should also mention this self-conditioning, right? We also had our XT, our X1 prediction from the previous step, always as input to our tensor field network. So in the self-conditioning thing. Now, what we do in our to include residue type predictions is that, yeah, we also first of all we have some invariant neural network rather than this some equivariant architecture, which we found to work a bit better. And then we predict an X1, an A1, which are now residue types, U types, our ground truth residue types, and we interpolate, right? We interpolate, okay, how does our discrete interpolation work? Well, the thing we do is we actually don't do this interpolation, and we don't use the AT as input. We only do this sort of recycling thing and only use the X1 prediction or the A1 prediction as input. And we train this, like we have this self-conditioned flow. We train this in exactly the same way as our as the self-conditioning setup that we described for the coordinates, and yeah, then can use this during inference. And that's the whole thing that's going on. There's no complicated discrete flow matching procedure or something going on. And with that, we can compare our ability to recover the residue types of some known binding sites. And we see that if we, for example, have a model that takes the ground truth ligand positions as input, then this helps quite a bit compared to a model that only takes the 2D ligand identities or the no information about the ligand as input and only can reason about the structure of the protein and basically does inverse folding. And with FlowSite, where we jointly generate the structure and the residue types, we nicely fall in between this uninformed no ligand or 2D ligand approach and something that has like some oracle method that has access to the ground truth ligand positions. But right here, this is important, and sort of if we now actually want to further tackle the enzyme design or no, just binding site design problem, here we're only looking at what is the percentage of residue types we're able to recover. And this is kind of not the metric that we're aiming for. And what we would want to optimize is the binding affinity that the ligand has to the protein. And we would want binding affinity as the evaluation metric. But then we're in this regime where, the binding affinity in general is just something very hard to predict. And if I had a good binding affinity predictor, I would probably write a paper on that. But here, then we have the additional complications that we don't know the side chain coordinates. Then estimating a binding affinity without side chain coordinates, there are again less tools for that. But yeah, I think important future work really is improving the evaluation here of what the designs are, right? With something like inverse folding, we have this nice designability metric that we can use by leveraging folding methods and looking at, is the refolded structure similar to the structure that we started from, and then it would be nice to sort of find an equivalent like this here. Yeah, and then with that, I want to say that there's also, if you're interested in binding site design, right, then there's now also these ligand MPNN papers from the Baker lab, and maybe something that overshadows this here a little bit is also RFAA diffusion, where Baker lab generates binding structures or generates proteins protein structures completely de novo that hopefully bind a small molecule. So you just have the small molecule as input, and then you generate a protein structure, and then the amino acids of this protein structure, right? Because you have the structure of the ligand with respect to the whole protein structure, those amino acids you then generate with protein MPNN, which now is called ligand MPNN because it also has messages to the ligand. Yeah, but here, where methods like FlowSite could maybe be promising is, or why they could still have value compared to something like this, is because they're not looking at the de novo design task where you're really generating a completely new protein, but it's like you already have a structure of a protein where you know, of a pocket where you know it's feasible that probably this could work for the small molecule or these small molecules. But I don't have the structure of the small molecules, and then using this prior knowledge and re-optimizing the pockets is where these tools could maybe still have some value in comparison. Okay, then do we have some more questions, or do we want to look at some videos to gain some intuition?
I think there's one question by Michael in the chat. For the ablation study, there was a sigma equal to 0 and sigma equal to 0.5 entry. That is the noise added when during the interpolation.
Yeah, exactly. That's the that's whether or not we add some additional noise to this X. And I was deceived. No, this is mean to say. I had a misconception that things would be correct if I added just constant noise everywhere along my interpolant. But then reading the stochastic interpolance paper again, they like have this in their stochastic interpolant. They always have basically no stochasticity in their interpolant at time 0 and time 1. And yeah, this is actually necessary if you really want to end up with the correct marginals of P1 and P0. Yeah, this is something I didn't know until 2 weeks ago. And if you want to hear more about that, you can watch the video from Michael Albergo talking about the stochastic interpolants. Yeah, this is the sigma was the noise.
Perfect. Michael, do you still have a question?
Oh, no, I figured it out. I had forgotten briefly what blind docking was, and that was
Okay, Prudencio, can you give me, and I'll do one last thing, if you give me rights to share my screen, and then
Sure.
I show a little video, which I think gives maybe a nice intuition on like also this question here that we had, right? About why do we need to do these multiple steps, and what actually happens during these multiple steps. Let me share. Which screen am I sharing? Are you seeing a paper? Ah, yes, you are.
Yes. Yeah, seeing your paper now.
Then let's go here. Hi. Look, and let's just look at an arbitrary protein, an arbitrary prediction that we'll make. Yeah, here, for example. And we always have our, let's see, what are these that we're looking at here? Yeah, so here what we're looking at is our X1 prediction. So this, the yellow thing is the ground truth structure, and here we're just have this harmonic flow structure prediction thing. And now the blue thing is a sample from the harmonic prior. And then this is the first X1 prediction, right? And you can really see this issue of stuff that is close together, of like symmetric things being squashed up in the first X1 prediction. And also here, if you have something symmetric, right, where you're not able to distinguish it, so to say, you have the symmetric things overlaying with each other. And now, as we in the next X1 prediction, things spread out already a lot, right? Here, this thing was spread out. Then let's look at the next 20. And here now all these rings almost are perfect already. And this is a sample from the test set, by the way. Yeah, a sample from the test set. And then we can maybe now look at this symmetry being resolved, and how over the course of our prediction this gets spread apart. And this is then the final X1 that we predict. And yeah, we're a little bit off, right? Here, for example, a little bit off. But in this case we were lucky, I think, and picked a sample here. Like this is randomly picked, not cherry picked, but we were lucky and have something good looking.